Hi everyone, we are so happy to have you, whether you are joining us here today or will be watching later from the Cure GABA-A YouTube channel. We ask that you please mute your mics if you have not done so already, and include any questions that you might have in the chat box below. We'll have time to answer questions at the end of the presentation. My name is Monica Joanna Alnacave, and I'm the founder and CEO of Cure GABA-A, a nonprofit organization with the sole focus to develop potential therapies for GABA-A variants by partnering with world-renowned researchers in GABA-A and the Organization of Patients for Clinical Trials. Today we welcome Dr. Alexander Rotenberg, who recently joined us at our in-person Cure GABA-A Scientific Roundtable that was held in conjunction with the American Epilepsy Society annual meeting this past year. He is also listed as one of our recommended physicians at Boston Children's Hospital. He is a prominent neurologist and epileptologist, serving as the director of the Neuromodulation Program within the Department of Neurology. He has received recognition for his work, including the 2016 Dreifuss-Penry Epilepsy Award from the American Academy of Neurology and presidency of the Greater Boston Epilepsy Society in 2015 to 2016. Dr. Rotenberg leads efforts to adapt non-invasive brain stimulation techniques like TMS for pediatric epilepsy patients, while also overseeing basic science experiments, translational applications in epilepsy in traumatic brain injury patients. His ongoing research includes clinical trial testing of efficacy of brain stimulation in drug-resistant seizures, studies in human brain plasticity, and epilepsy. I'm handing it off to you, Dr. Alexander Rotenberg. Please help us understand the phenotypic manifestations observed in individuals with GABA-A variant mutations.
Very much, Monica. And thank you to the audience in advance. Thanks for the opportunity to talk about transcranial magnetic stimulation, which is a way for us to interrogate the motor cortex. And let me, by way of introduction, talk a little bit about TMS, which the group may have heard about. TMS is not new anymore. It's a technology that's been around for about 30 years, and you can see articles in the New York Times and Boston Globe and announcements from the FDA about approvals of this technology for a range of therapeutic purposes. In other words, there's lots of syndromes that are quite prevalent, like major depression and obsessive compulsive disorder, maybe even epilepsy, that can respond to transcranial magnetic stimulation. But what I want to talk about now is a less popularized, but from my perspective, more practical in the GABA-A variant community use of TMS, and that is to use TMS as a diagnostic tool. To back up a little bit, let me talk about the basic principles of transcranial magnetic stimulation. Even though it has the word magnetic in it, the M is actually not what's doing the brain stimulation. TMS is based on the principle of electromagnetic induction, which was, you might remember from high school physics, discovered by Michael Faraday. And it's the principle where if you pass electrical current in one conductor, let's say a wire coil, in another conductor that may not be even touching it, you could appreciate the pickup of that electrical current. And it's the principle by which a electric field generates a magnetic field, and that magnetic field then induces an electric field in a nearby conductor. This is the way power plants work, for example, or transformers or other things. As a quick tourist aside, if you're in London and you're really cool, you can skip the boring parts like Big Ben and Parliament and go straight to the Michael Faraday Museum, where you can see some of these original artifacts. And he was actually a very prolific inventor, so it's a site worth visiting. But it's actually from these artifacts that we have these devices. And so the microfaraday electromagnetic ring was embodied in the 1980s into a transcranial magnetic stimulator. This is the first of these devices being used by Dr. Anthony Barker, who invented this instrument. And you can see what the operator is doing in this picture. And all the devices since then have looked very similar. There is a capacitor that charges to about 10,000 volts. That charge is relieved through a steel cable that connects to an electromagnet. That magnet makes a strong magnetic field, and just like in Michael Faraday's coil, makes an electric field everywhere around it, including in the brain. You can actually see that the operator here has the stimulator over her right hemisphere on the right side of her head, and in her left hand, she has an electrode on her palm. That electrode is there to record a signal from the muscles of her hand. So the way TMS works is that it generates a strong electric current in the TMS coil that makes a strong magnetic field. That magnetic field that makes electricity everywhere around it, including in the brain if we position it next to the scalp. Now, the part of the brain that talks back to us the fastest, if we take the stimulating coil and put it next to the scalp, is the motor cortex. And that's kind of intuitive, because if we stimulate the motor system, something moves on the opposite side of the body. And we can quantify how strong that movement is by putting surface electrodes on the skin of the participant. And that gives us something very valuable in electrophysiology. At least for us, electrophysiology nerds, this is something very important. It's an input-output curve. We know how much energy went into the brain, and we know how much energy came out through the muscle. And so by this technique that's non-invasive, relatively focal, and painless, we can record the motor evoked potential, the MEP, and we can put a number on it. And the technique to do that, as you can imagine, in diseases like epilepsy, can give us very valuable measures of brain excitation and brain inhibition. So these devices have been around since 1984. This is Tony Barker and his participant in the first embodiment of this device. This is Gabby and Natasha, our research assistant and secretary, in a more modern device, but essentially they all have the same components. There have been some innovations recently. One of them you can see here, which is the capacity for the operator to look at a patient's MRI and deliver the electrical current relatively precisely to a part of the patient's brain, and some advances in the geometry of this magnetic coil, where, notwithstanding that these coils can be about the size of the palm of a hand, depending on the configuration of the electrical current inside the coil, and this, what I'm showing now, is something called an Archimedes spiral in a figure-of-8 arrangement. The current can sum at the center and fall off at the periphery, so that the real stimulation is happening only in about a fingertip's volume of the brain. So the plain English here is that TMS is a way for us to non-invasively and very focally stimulate the cortex. And if we do this again over the motor part of the brain, that stimulation gives us an input-output curve. That input-output curve can vary depending on the disease or the drug, and can give us a sense of whether or not brain inhibition is normal or abnormal, or whether brain excitation is normal or abnormal. I'll just quickly mention a couple of the new bells and whistles that are there in modern devices. One of them is frameless stereotaxy. So this is, again, that capacity to register the patient to a stereo camera, stereo with 2 lenses, so you can see in 3 dimensions. And that camera is then connected to a computer, and that computer can then identify for the operator where the patient's brain is in 3-dimensional space, and where the electrical charge is landing on the patient's brain, so that the operator can visualize the brain anatomy and deliver the charge selectively to the motor system. And again, for purposes of this talk, I'll bias very strongly to talking about TMS as a way to stimulate the motor cortex. And one thing to keep in mind is that the motor part of the brain is so well connected to the rest of the brain that if we stimulate the brain and record the motor output, but putting electrodes on a muscle whose brain representation we're targeting, we can learn something about the rest of the brain. In other words, the motor cortex is so well connected to the rest of the brain that depending on what the rest of the brain is doing, the motor cortex will give us a readout on whether or not that brain has been abnormally excited or abnormally depressed or otherwise. And I hope that makes sense. And if not, please pause me. So TMS, again, is something that's non-invasive, relatively painless. It's actually available across ages. In my day job as a pediatric neurologist, as you can imagine, we administer all sorts of things to children. And this is actually something that's very well tolerated by participants, even in the very delicate pediatric age category. So again, the purpose of any of these experiments is to stimulate the motor cortex and to record by surface electrodes what the muscle did in response. Now, any kind of brain stimulation, including TMS, can be roughly divided into three types of stimulation protocols. One is a single pulse stimulation. So we stimulate once, and we see what the brain did in response to that single stimulus. And again, for us, we stimulate the motor system, and we see what the brain did by looking at the motor output in a hand muscle. It could be any other muscle, but somewhat arbitrarily, but in part because the hand takes up so much real estate in the brain, we're looking at hand muscles. Another kind of interesting protocol is to stimulate in pairs. And I'll talk about this in detail, but if we stimulate the brain in closely spaced pairs, the first stimulus changes the brain's response to the second stimulus. And it becomes a way for us to sort out whether or not cortical inhibition, which is reliably triggered by a stimulus to the brain, is affecting the response to the second stimulus. And then there's what's used in therapeutic interventions, stimulating the brain over and over and over again for purposes of changing brain excitability. And this is how TMS devices have been used and approved by the FDA for treatments of things like major depression. In other words, these devices take advantage of a fundamental property of the brain called synaptic plasticity, where if we deliver trains of electrical stimulation, we can change how excitable the brain is. So I will try to cycle through these three in the rest of the talk. And that's an okay way, from my perspective, to organize how we think about TMS, which is what can we do with single pulse stimulation, paired pulse stimulation, and repetitive stimulation, and what can we learn about it? So single pulse stimulation we use all the time for purposes of mapping the motor system before some kind of surgery. In our line of work, it's often epilepsy surgery in children. So here's an example of what we do every week in our TMS clinic. We have a participant. We image that participant's brain. We deliver a stimulus, and in this case, the stimulus is where these red and blue arrows meet. And these dots correspond to where we could produce one of these. This is the motor evoked potential, in this case, from the abductor pollicis brevis muscle, which is the muscle that moves our thumb. And what we can see here in the right panel is 6 channels of electrical signal from 6 muscles, but only one of them gives us this deflection. This corresponds to the motor evoked potential from the muscle that governs thumb movement in this patient's right hand with stimulation of the left hemisphere. The other muscles, which are connected to the foot or the shoulder or otherwise, or to the other side of the body, are giving us only artifact, but no motor evoked potential. The product of one of these sessions is something like this: a map of where is this patient's hand motor representation in this patient's brain. We can do this for the hand. We can do something similar for the foot, for the tibialis anterior muscle. And then the question is, what do we do with it? What's so good about it? Well, most of what we do with this is something like this in another patient. So in this case, here is a patient. You can see, without being a neurologist and neurosurgeon, this patient's tumor, it's a low-grade glioma in the dominant hemisphere for a patient who, this was an adolescent, was learning to drive, and could not afford to lose function in the right side of the body. So we were able to map the hand representation by doing the kind of stimulation that I just showed, and map the foot representation, and then identify for our tumor surgeon a corridor that wouldn't interrupt either foot or hand function that the surgeon could use to take out this tumor. So this is the use case of single pulse TMS combined with electromyography, that recording of the electrical signal from the muscle. Now, in making these motor maps of the brain, we can do something else very interesting and a little more relevant to our discussion here, a discussion of cortical excitation and inhibition. So while we're mapping the motor cortex, we are obtaining these data. This waveform has a couple of things that we can quantify. One of them is its amplitude. This waveform happens to be 627 microvolts in size. And if we can quantify this waveform, then we can identify how easy is it to produce something of a fixed size. And so by having this as an output, we can have actually a pretty broad range of measures of cortical excitability. So these red arrows represent where the stimulus is and what pattern. And I won't bore you with the details, but we can use the size and the latency and the character of these motor responses to tell us how excitable the brain is. And let me give you two examples of these that I think are most closely related to what we can expect to see in some of our patients. One of them is this concept of a motor threshold. It's exactly what it sounds like. It's the threshold for us to obtain this motor response by stimulating the brain. So you can see here we have units 33%, 34%, 36%. MO abbreviation in this case stands for machine output. How much of that capacitor do we need to discharge to get the brain to respond to us with a motor signal coming out of the hand? So we can see at 33%, nothing happens. At 34%, nothing happens. Something happens at 36%. So we say the threshold is probably at 35% for this patient. And as you can imagine, the more excitable is the brain, the lower is this threshold. The less excitable is the brain, the higher is this threshold. This, for us, in a walking, talking human, is extremely valuable in neurophysiology. We always want to know how excitable is the patient's brain in a baseline state, in a disease state, let's say, and then we want to know whether or not our intervention, whatever that is, it could be a gene, it could be a drug, it could be a stimulation protocol, has changed that excitability. And there's a predictable pharmacology to this motor threshold, so that threshold is reliably increased, for example, by common voltage-gated sodium channel drugs, oxcarbazepine, phenytoin, lamotrigine, etc. And here's an example of how to use this in epilepsy research. There's a new drug potentially coming on the market. Its generic name right now is Xenon 1101. It happens to be a voltage-gated potassium channel opener. And how do we know that this drug has affected the brain in a healthy participant? We would always want to do phase 1 trials in somebody who does not have epilepsy. In epilepsy, we could see if the seizures get better. But in a healthy participant, we'd want to know, has drug X hit the brain? And one way to know it is to measure on the y-axis this motor threshold, on the x-axis the concentration of drug, and to see whether or not the resting motor threshold increases, as this line shows, as the concentration of drug increases. So one cool way to use this form of non-invasive stimulation, and just to remind the group, what we're doing here is we're using non-invasive brain stimulation to put an input into the brain and get an output from a hand muscle, is to apply it while administering an intervention. It could be a drug, it could be a gene therapy or otherwise, to ask whether or not this measure of excitability has changed in the direction that we want it to change. So if this drug is anti-epileptic, we would predict that this change would be toward less cortical excitability or a higher resting motor threshold. That's what this graphic shows. Now, there's another cool finding with this same measure, which is not telling us whether or not the drug has reached the brain, but whether or not the drug has changed the brain in the way we want it to change in somebody with epilepsy. So let me walk you through this old experiment. It's already almost a decade and a half old, and it's really cool. It comes out of a group in Australia that studied patients with either idiopathic generalized epilepsy, that the abbreviation IGE, or focal epilepsy. And then after this patient was diagnosed with epilepsy, these patients were given a drug. And they were sorted into two groups: patients whose seizures stopped after they were given a drug, and patients whose seizures continued. And then the gray and pink boxes here correspond to the resting motor threshold before the drug was given and after the drug was given. And here is one example. What I'm circling here with the highlighter is what they found that looked most favorable, which is that in patients whose seizures went away after a drug was administered, the post-drug resting motor threshold on the y-axis increased, as you would expect. If you want the brain to become less excitable, you would want the threshold for activating it to increase. Interestingly, in patients whose seizures continued with the same dose of drug, there was no change in the resting motor threshold. So another cool outcome by measuring the motor cortex excitability by TMS is that not only could you identify whether or not a drug has reached the brain, you could also potentially measure whether somebody's disease has changed. pursuant to the administration of a drug or a gene therapy or otherwise. Now, let me show you something else that's really interesting about the motor system. The motor system is very well connected to the rest of the brain. I know I keep saying this over and over again. And the idea here that I'm trying to transmit to the audience, I think, is that we can use the motor system as a surrogate to tell us how the rest of the brain is doing. And here's something interesting that the motor system does. On the y-axis is that same motor threshold. How much energy do we need to get something to move on the opposite side of the body? And what we see actually is that the threshold declines with age. The brain actually, as far as TMS and the motor system is concerned, becomes a little more excitable as we age. It stabilizes a little bit by adolescence, but you can see through childhood, it becomes easier and easier for us to activate the brain by TMS. And these are published data from Laura Saisonen in Finland. And we've complemented these data by combining our own data from the laboratory with Sandra Krieg, a neurosurgeon from Munich who runs a very productive TMS lab, where we could see the resting motor threshold across ages. So we took our pediatric cohort and the Munich cohort of adults. We combined them together and we see something very interesting. Through about adolescence until the teens, the resting motor threshold, that's the y-axis value, steadily declines, and then from the 20s to about age 90, stays very stable. Now, this tells us something very interesting about the motor system and maturation, which is that if we have a measure that changes with age and then stops changing, what we have is not just a measure of maturation, but what we have is this inflection point. This inflection point corresponds to something potentially valuable to us. It corresponds to maturity. In other words, something in the brain can change with age, and it'll change, and then it'll stop changing. And after it stops changing, we can say it's not just maturing, it's matured. It's reached maturity. Imagine your shoe size. It'll change up to a certain age, then stop changing. We can say at that point, your shoe size is matured. The same thing we can say about the motor threshold. It'll change with age and then stop changing. And when it stops changing, we have something very interesting. We have maturity. Now, why is that cool? It's cool because TMS and the motor system can tell us something about maturation in the normal brain and maybe even about maturation in the abnormal brain. And this is something that's so far a solution waiting for a problem, so to speak. But let me show you something else here, which is a graph from our data, looking at patients with epilepsy, and the filled and empty circles correspond to boys and girls. I forget which is which, to be honest. But what you could see actually is a trend here, and that's what this trend line shows, which is the higher is the verbal IQ on the x-axis, the lower is this resting motor threshold. The units here are volts per meter. Now, why is this interesting to us? Because if something is measuring maturity and something can identify for us a point of maturation, and the IQ ultimately is a ratio of how mature is one's response to a series of tasks versus one's chronological age, then TMS can even tell us something quite sophisticated, which is how mature is somebody's brain physiologically relative to their age. And again, this is my plug for how cool the motor cortex is, because even though our caricature impression from high school through medical school is that the motor cortex is this very primitive structure that just talks to the spinal cord and gets our limbs to move but doesn't do much else. Realistically, it's actually telling us about relatively sophisticated functions in the brain, even something as sophisticated as IQ. So let me just stay on this theme and tell you about another TMS protocol, which can measure for us GABAergic inhibition. And this is paired pulse stimulation of the motor cortex. So in paired pulse stimulation, like it sounds, we have two stimuli. The first one is called the conditioning stimulus. The second one is called the test stimulus. The brain's response to the first conditioning stimulus determines what it does to the second stimulus. That is, that we stimulate the brain once, and after we've stimulated a volume of cerebral cortex, we've activated all sorts of excitatory neurons. Now, almost every time that we activate excitatory neurons, they're going to be connected to a network of regional inhibitory GABAergic interneurons. Now, those neurons are going to be activated. So by the time the second stimulus lands, we will have a different response and usually a dampened response to the second test stimulus that was provided for us by the conditioning stimulus. I hope that makes sense. So bottom line is that if we do pairs of stimulation, the response to the second stimulus is often not the same as it would be to the first. And depending on the interval between these two stimuli, it could be predictably smaller. So that if we record, for example, 2 consecutive responses from an individual, in one, the response to a single stimulus, in the other, you can see the 2 stimulus artifacts here, in response to a pair stimulus, the paired response is predictably smaller. And this is excellent work from a group of Don Gilbert and Cincinnati Children's that have been looking at this kind of paired pulse GABAergic inhibition in a range of diseases, including relatively sophisticated diseases as far as brain function goes, like attention deficit. And what Don Gilbert has found in his lab is, again, if we plot on the x-axis here, short interval intracortical inhibition, which, let me back up a couple of slides, corresponds to this phenomenon, where if we have 2 stimuli spaced only 2 milliseconds apart, the response to the test stimulus is predictably smaller than the response to the single stimulus. And it's smaller because the first stimulus activates a whole bunch of GABAergic surround inhibitory interneurons, so that by the time the test stimulus lands to that same cortex, it can only produce a small response because all of that active GABAergic signaling. And if we look at the severity on the x-axis of the patient's ADHD symptoms, and on the y-axis of the magnitude to which that second stimulus has been inhibited, so the higher the y value, the less inhibition, we could see that the more severe the symptoms, the less there is inhibition of that stimulus. So let me just back up again, just to say that what we're looking at on that x-axis is the ratio of this stimulus to this stimulus. This will be the numerator, this will be a denominator. The closer it is to a ratio of 1, the less inhibition there is. The smaller the value, the more inhibition there is. And what we could see is that by, again, stimulating this primitive part of the brain, the motor cortex, that doesn't do much else than talk to the spinal cord, we can get a readout on something very sophisticated, which is how much GABAergic inhibition there is, and whether or not that GABAergic inhibition is corresponding to something that can be measured in a patient's behavior, like the magnitude of the patient's attention deficit. Now, there's something interesting here, which actually these authors didn't comment on, I think, in their paper, which is if we look at a value of 1.0, that already means that the size of the response to the single stimulus and the paired stimulus is identical. Yeah, a ratio of 1 means the numerator and denominator are the same. If that's the case, then there are some values here that are outliers. They're not just not enough inhibition, they are actually lost inhibition, and they are showing facilitation, which is actually very interesting for us. This is already showing us something else that the motor system and TMS can tell us, which is a qualitative difference in the way GABAergic signals are processed in some patients. So along these lines, let me quickly point to a disease that we're studying in the laboratory right now. This is a very rare genetic disorder of GABA signaling called succinic semialdehyde dehydrogenase deficiency. It's a disorder of GABA catabolism, which is the degradation of the GABA molecule in the mitochondria, and the compromise in GABA degradation is due to the absence of this enzyme, succinic semialdehyde dehydrogenase deficiency. It's a very rare disorder, only several hundred in the United States. And it's characterized, as you can imagine, if you're not degrading GABA, by excess GABA. Yet, even though these patients have an excess of the major inhibitory neurotransmitter, they're not asleep all day long, and on the contrary, they are agitated and insomnic, and many of them are epileptic. And what's happening in this patient population at the molecular level, at least what we can see from PET imaging in humans and from mouse studies that we're doing in the laboratory, is that in response to this GABA excess, there is compensatory downregulation of the GABA-A receptors. With enough downregulation of the GABA-A receptors, if this compensatory response overshoots, then we actually have a net loss in GABA inhibitory tone. If that's the case, one question for TMS is, can we measure it? And if we can measure it, can we measure it by this paired pulse stimulation protocol, where we deliver a conditioning stimulus followed by a test stimulus. And again, in a simplistic version of this paired pulse TMS protocol, there is a response to the first stimulus and then a response to the second stimulus. And the response to the second is never as big as it is to the first in a healthy brain, because the first one activates all this network, a GABA-mediated cortical inhibition that dampens the response to the second stimulus. So if we use this protocol in patients with succinic semialdehyde dehydrogenase deficiency, what we see again is something fascinating. We have 2 or 3 categories of the response to this paired pulse stimulation in succinic semialdehyde dehydrogenase deficiency, which is that some patients respond normally, but the majority of patients respond either with an absence of inhibition or with this paradoxical facilitation where GABA signaling, at least by the way we're testing it by TMS, is doing something else than we expect it to. It's actually, instead of net depressing the network, it's actually facilitating the network. And again, this gives us not just a quantitative difference from healthy control, but a qualitative difference from healthy, and even within this disease, qualitative distinctions among subpopulations. So for us, this is actually a very valuable biomarker because it may distinguish healthy from unhealthy. And even among patients with a syndrome, it can distinguish for subtypes and maybe even predict how those subtypes will respond to an intervention. So again, just to reiterate, GABAergic cortical inhibition can be measured effectively in humans by paired pulse transcranial magnetic stimulation, 2 consecutive stimuli delivered to the motor cortex, where we measure the size of the motor response to these 2 stimuli and then plot them on a graph. Now, it turns out that we can do the same thing in rats that we can do in humans. And realistically, the trick here is actually not that hard. We can, in the laboratory, expose an animal to TMS and get 2 consecutive motor responses, and the size of the second is never as big as it is to the first. Now, we can do this in humans, so the question is, what's so good about doing this in animals? And one reason to do any of this work in animals is that we can model human disease in the laboratory. We've modeled a number of human diseases in our laboratory. Let me tell you the story about one of those diseases. This is acquired epilepsy. About 20% of all epilepsy is acquired after some kind of brain injury. It could be a stroke, it could be infection, it could be trauma. It turns out in young adults, the most common way to acquire epilepsy is with head injury. It's overwhelmingly in a civilian population, motor vehicle accidents. Now, the unfortunate natural experiment that happens in post-traumatic epilepsy is that there's an injury. It's often to a young and healthy brain. And then for months, nothing happens that we can see with the naked eye. But while there's no epilepsy after the injury for several months to up to a couple of years, there's necessarily something happening in the brain, because at the end of this prolonged period of months to maybe even years, epilepsy happens. And during this period, the brain is undergoing changes, and some of these changes are well described. They involve inflammation and oxidative stress and neuronal loss, and they are completely invisible to the naked eye. But in the animal laboratory, we can maybe see them. So in the animal laboratory, we can model the human disease. We can deliver an injury to the animal cortex, and I won't belabor how this device works, but we can cause a contusion to the motor cortex. The bright spots here are dying neurons. We can cause cell death, and 6 weeks later, we can see seizures on EEG. So the interesting question we can ask in the animal laboratory, where we have a scenario where it takes about 6 weeks to find any kind of seizure, is could we measure something in the brain that's happening over these 6 weeks? And could we measure it by this technique where in humans and in animals, we can stimulate the brain twice, measure the size of 2 consecutive responses of the motor system, and ask how much smaller is the second response than the first? And Henry and Sean, our scientists in the laboratory, did exactly that. They injured an animal and obtained 2 consecutive responses week after their injury. And you can already see in this cartoon, in the animal with traumatic brain injury, the second response grows in size. And it's growing in size because of a hypothesis that we're testing that turned out to be correct, which is that we think that there's loss of inhibitory interneurons in the acquired epilepsy. Now, in humans, we can't see that with the naked eye, but in animals, we could see it, and we might be able to see it in humans sometime soon. So again, the experiment that Sean and Henry ran was to injure animals, to give them either a sham injury or a real injury, and on the y-axis plot the ratio of these two responses. So the closer that ratio is to 1, the more equal they are. The more equal they are, that means the less inhibited is the second response. So we can injure animals every day and every week ask after their injury, how's your cortical inhibition, how's your cortical inhibition, et cetera, et cetera. And what we can see is in the injured group, we have a gradual loss of intracortical inhibition over time. That's what the open circles are showing us. Now, we can look at the brain, that's the luxury that we have in the animal laboratory, and see that there's not much happening if we just mark every neuron for the microscope. But if we zoom in on subpopulations of neurons, and especially on GABAergic interneurons, and among those GABAergic interneurons, on the most common kind of GABAergic interneuron, which is the parvalbumin positive inhibitory interneuron, which is especially susceptible to injury, what we can see over time, and that's what's shown in these micrographs, is that around the lesion, there's gradual loss of this red marking. And this corresponds to a gradual loss of cortical inhibition. So the value of this experiment for translational purposes is that in humans, we can imagine something similar happening. That is, that we can perhaps identify a person with an injury, and before they develop seizures, ask at regular intervals, how's your cortical inhibition? How's your cortical inhibition? And not just how's your cortical inhibition, but specifically, how is your GABAergic interneuron-mediated cortical inhibition? And maybe use TMS delivered to the motor system as a non-invasive measure of how intact or how compromised is this patient's GABAergic inhibition. So we think we have actually a solution to this GABAergic inhibition, which is treatment of the astrocyte. And I won't belabor the biochemistry here, but I'll just say that we think that the reason why we're losing these very fragile GABAergic interneurons is because after injury, we lose from the astrocytes the way for us not to deal with GABA, but the way for us to deal with glutamate, the major excitatory neurotransmitter. Because if we have a loss of this astrocytic glutamate transporter, we have an excessive glutamate. So if we have a loss of this protein, we have an excessive glutamate. The excessive glutamate, I won't belabor the point, causes any number of things to happen downstream, including actually a loss of the material that we need to make the major antioxidant in the body called glutathione. And if we have that loss of glutathione, we then have injury to the cells that are most vulnerable to oxidative stress, which happen to be our GABAergic interneurons. Now, it so happens that we can potentially, at least in animals, fix this loss of glutamate transporter and this gain of glutamate excitotoxicity, which injures preferentially GABAergic interneurons, with a common antibiotic. It turns out that for some crazy reason, beta-lactam antibiotic, penicillin and its cousins, in this case, it's the cousin of penicillin called ceftriaxone, can restore the amount of this glutamate transporter, and in animals can suppress seizures after brain injury. And that's what's shown on the y-axis here in saline and ceftriaxone-treated rats. So thank you for allowing me to go down this small molecular rabbit hole. And the practical question is, so what? Why is this interesting to a TMS lab? And the reason why it's interesting to a TMS lab is because if TMS, which we can do in rats and we can do in humans, can measure for us the gradual change in cortical excitability as a patient progresses from an insult, and an insult could be something infectious or something traumatic or a stroke, or maybe even a genetic load. But as the patient progresses from the insult to epilepsy, we can potentially, by TMS, measure their cortical inhibition. And that's exactly what one of our investigators, Mustafa Hamid, did in the laboratory. He measured by TMS in rats these two responses to the conditioning and test stimulus, and identified that over these weeks that it takes for epilepsy to develop in animals, the animals treated with ceftriaxone maintained their cortical inhibition on the y-axis better than the animals treated with saline. So for the first time, we now have a biomarker, not of epilepsy, but of epileptogenesis. And not just a marker of epileptogenesis, but a marker that corresponds to how intact is our parvalbumin positive GABAergic inhibitory interneuron system. So that's the value of doing TMS as a diagnostic measure. And I'll just blast through for a second about TMS as a therapeutic, which is this repetitive TMS protocol that, if delivered in low frequencies to a seizure focus only in some studies and not all studies can suppress seizure frequency. This you might have already seen. This is a well-publicized study from Beijing where patients delivered sham stimulation, did not have a change in their seizure frequency on the y-axis, and patients delivered repetitive TMS, which in some frequencies delivered to the motor cortex may reduce cortical excitation and may work the same way around a seizure focus, had some relief of their seizures, but the relief was not complete. We've done the same with other kinds of TMS coil. This is a study of temporal lobe epilepsy. And we do what we always do in epilepsy. We made good-looking bar graphs. This is something we're often self-congratulatory for. We have no change in a sham condition, some meaningful change in a variant condition. But if we zoom in on the data, this is what we see. We look at these patients on the y-axis, daily seizures. These are what the bars correspond to. We see they pause during 2 weeks of TMS and gradually they relapse. This would make for a very good-looking bar graph, but this patient still can't drive. So our treatment and our success with stimulation looks a lot like this. And this doesn't matter, in fact, if this is TMS or VNS or RNS or DBS. The common denominator for any kind of brain stimulation in epilepsy is that our success is meaningful but incomplete. So these patients may pause their seizures, and on the y-axis is seizure counts per day. They will relapse, not completely to their baseline, but nonetheless, they relapse. So the question for us is, how do we improve? And we improve simply by bouncing back and forth between the animal and the human laboratory. We can reduce some of these preparations to the animal lab, trigger seizures how we want. In this case, it's an animal EEG with a trigger by a chemical convulsant, and then march our way to the parameter space, for example, to identify, does a quarter hertz stimulation work, or half hertz stimulation, or 3/4 hertz stimulation, and find what works best in the animal model, and then forward it back to the clinical arena. So this kind of back and forth is something we can do in non-invasive brain stimulation, and in fact, in many kinds of brain stimulation, I think better in many other fields, in part because the brain stimulation gizmos, TMS and other forms of brain stimulation, have been deployed in the clinical arena far ahead of the basic science that supports them, which means that we can have devices that are already out there being used on patients that have proven safety profiles. And if we forward data from our laboratory, we may actually improve how they're being used in patients. So that's the quick summary of TMS and how we may use it mostly as a diagnostic tool, but potentially as a therapeutic tool in epilepsy. And I'll just close with this historic slide, which shows Jacques-Arsène d'Arsenval, who's a French physiologist who worked mostly in the tail end of the 19th century, beginning of the 20th, who's the first to identify that induced electromagnetic fields can affect the brain. And what they did is they put a participant into this electromagnetic coil, and the participant felt dizzy and saw flashing lights, and they said, aha, you could induce electromagnetic current in somebody's brain, and it's meaningful. But it's not the physiology that's interesting in this photo. It's what I've heard about who these people are. And the cool thing about this photo is that the guy here running the equipment is the laboratory technician. The person who's putting his head into this novel device is the laboratory postdoc. And the guy who's sitting here off to the side and taking credit for everything is the lab principal investigator. So since I've been showing our laboratory work, let me assure you that I've done none of it. The entirety of our laboratory work is really due to these young investigators and their loved ones who let them spend weekends in the lab. And so everything you liked about this talk is their credit. Everything else is mine. So thank you very much.
Thank you so much for your time, Dr. Rotenberg.
Oh,
that presentation was absolutely fantastic. Very informative and easy to follow.
As you know, Monica, and whoever doesn't know me, I am available. My email is easy. My phone number is accessible. Call and ask any questions.
And you are listed on our website as a recommended physician.
Sounds
so
good.
If anyone is interested in the Boston area, they can reach out to you. They can reach out to us. We can put them in contact with you. See you soon, Dr. Rotenberg.
Bye, everyone.
Bye.